Hello everyone, this is Masood Ahmed. Today, the topic of my presentation is Pharmacogenomics, finding the right medication for the right person. So, in today's presentation, we will cover the motivation behind our topic. We will discuss a case study and talk about LL combination and ultra rapid metabolizers in order to reach to the conclusion of the case study. Then I will mention some of the common genetic testing that happens today and then talk about what are the current use of pharmacogenomics and what will happen or what might happen in the future. So, before going into the depth of the topic, let's consider a simple example. Imagine that you have just given birth to a healthy boy and you are now a new mother. Unfortunately, you now have crippling postpartum pain as a result of the delivery. Your doctor prescribes you a painkiller which helps you to but also makes you feel drowsy, tired, and sometimes constipated. At the same time, you notice that your son isn't breastfeeding very well. The doctor reassures you that everything's fine and recommends you to freeze and store your excess breast milk for convenience. And you go home relieved. One day, your child doesn't wake up. He is not breathing and his skin is grey. Frantically, you call 999. But it's too late. Authorities arrive, an autopsy is done, extremely high levels of morphine are found in your son's blood. Infanticide is considered and your life is in shambles. This example that I shared right now is based on a true case study which was published in Lancet back in 2006. In that case, the culprit ended up being codeine, an opioid analgesic. Codeine is found in Tylenol number 3, a painkiller prescribed to many new mothers to relieve postpartum pain. Yet, the vast majority of these breastfeeding mothers have healthy babies. Why is this so? We'll explore this in this presentation on pharmacogenomics, and we will talk about how our genes respond to various medications and how we can find the right medication for the right person. So, in our body, we have a catalyst kind of a thing called enzyme. Enzymes break down and structurally alter the drugs once they enter our bodies, either by activating them or deactivating them. So, in short, an enzyme works on a drug or a or something called substrate and converts them into a product. For our case study, we will focus on P450 enzyme family. The cytochrome P450 enzyme family is the most well-known group of drug metabolizing enzymes. There are more than 50 distinct CYP450 isozymes. However, for the purpose of this presentation, due to time limit constraints, we will only focus on CYP2D6. The CYP2D6 gene sequence varies between people. These genetic variants or alleles result in slightly structural changes in CYP2D6 enzyme. These structural changes may alter how effective the enzyme is at metabolizing the drugs. These enzymes can be broadly categorized into 3 categories: normal function, decreased function, and non-functional. The normal function variant metabolizes the drug quickly. The decreased function variant metabolizes the drug slowly, and the non-functional variant does not metabolize the drug. So, each person has 2 CYP2D6 alleles since we receive a copy of each gene from each parent, one from a father and one from a mother. Then, depending on the combination of CYP2D6 alleles a person inherits, they can be categorized as rapid, which means normal, or they can be categorized as intermediate, or they can be categorized as poor metabolizers. A person who is a rapid metabolizer will have at least one functional allele. However, an intermediate metabolizer will have a decreased function and a non-functional allele. And a poor metabolizer will have two non-functional alleles. You can see all the possible combinations here, where you can observe that the majority of people are rapid metabolizers, around 77 to 92%. An ultra-rapid metabolizer is a category that can be occasionally applied to a person. In the graph, you can observe that a person being an ultra-rapid metabolizer has a probability of 1 to 2%, which is extremely low. These people have 3 normal alleles of CYP2D6 rather than 2. The reason for this is the gene duplication. Because of this, they metabolize the drugs extremely rapidly and efficiently, since they have more enzymes. This change in metabolism can impact a drug's potency and toxicity. Then drugs can either be activated or inactivated after they are metabolized, and as ultra rapid metabolizers have 3 alleles, they metabolize the drug more quickly. Now consider that if all the metabolizer types were given the same codeine dose, an ultra-rapid metabolizer would convert way too much codeine into morphine, which theoretically should be receiving a low dose to prevent overdose. It turned out the mother in the case study that we are studying right now, was an ultra-rapid metabolizer, and excess morphine entered her breast milk into her son because of her being ultra-rapid metabolizer, and this likely caused the overdose of morphine. And this is supported by the case study and autopsy that was done, in which high levels of morphine were found in her frozen breast milk as well. So the conclusion of this case study isn't that the codeine is a bad drug. It is quite an effective painkiller if used on the right people, and can be lethal on the wrong people. Now the real question is, how can we find the right medication for the right person? Well, the answer to this lies in the field of pharmacogenomics. It is genetic testing. So what is genetic testing? You might be wondering right now. Genetic testing or DNA testing looks for changes in one or more genes that may cause a genetic variation or a condition. The result of this testing can provide information on future health risks and can also change the way you are medically treated. There are 3 possible results of genetic testings. One is a positive result, in which the genetic change will inform an increased risk of medical problems. The second, you might have guessed, is a negative result, where no important changes were found. Lastly, there are unclear results, where results are found but it is unclear about what they mean. Here, in this slide, is a brief description of different types of genetic testing that are being done in today's world. One type of genetic testing is called carrier testing. This type of DNA testing is used to identify people who carry one copy of a gene mutation, which, when present in 2 copies, can cause a genetic disorder. The second type of genetic testing is prenatal testing. This DNA testing is to identify the changes in fetuses' genes or chromosomes before birth, so that any disorders or disease can be prevented before the birth. The third type of genetic testing is pre-implantation testing. It is a specialized technique that can reduce the risk of having a child with a particular genetic or a chromosomal disorder. The fourth type of genetic testing is predictive and presymptomatic testing. This type of testing is used to detect gene mutations associated with disorders and appears after birth, often later in life. Now, let's dive in and discuss where pharmacogenomics is being used these days. So, I believe that it would not be wrong to say that with the emergence of the field of pharmacogenomics, the field of medicine has been revolutionized and we are now able to save more human lives. In this slide, you can see two different areas where pharmacogenomics have helped. For example, in the field of HIV, genetic testing has dramatically reduced the number of people suffering from the side effects caused by the drug treatment of HIV. Abacavir is one of the drugs that is used in the treatment of HIV, but with its treatment, it is found that around 5 to 8% of people using this specific drug are prone to side effects such as rash, fatigue, and diarrhea. This is because their immune system produces exaggerated responses to the drug, which can even be considered as allergy. This is because of the gene variant called HLA-B5701. This gene variant produces a hypersensitivity reaction to abacavir. These days, people are screened out for HLA-B5701 and treated accordingly to minimize the side effects of abacavir drug, all thanks to pharmacogenomics. Similarly, we have another example related to rheumatoid arthritis. People with rheumatoid arthritis have a genetic test to see if azathioprine will be an effective treatment for them or not. This is because azathioprine is an immunosuppressant, which in some patients, when treated for rheumatoid arthritis, does not get activated. So this unconverted azathioprine builds up in their bone marrow, which in turn kills white blood cells, leaving the patient vulnerable to infections. The reason for this is that some variants of gene encoding of thioprine S-methyl transferase (PPMT) are unable to convert properly. These days, before treatment, patients are checked for what gene encoding they have and then they are treated accordingly. So from these two examples, we have just seen how pharmacogenomics have changed the way the people are treated. Finally, with such an improvement in technology and research, we might expect that in the coming future, we may have developed genetic therapies to cure some illnesses without even the use of conventional drugs. But until that occurs, pharmacogenomics offers very significant opportunities to allow conventional medications or medicines to be used in a more effective fashion. In addition, pharmacogenomics will allow drug companies to revisit their drug catalogs to find medicines that were too dangerous to license for the use of general population, and which, after the introduction of pharmacogenomics techniques, may be safely now prescribed to those having a particular DNA profile. It may also deeply be the case that the best treatments for many people for many different conditions are not currently on the market simply because they are lethal for those with a particular genetic makeup. Pharmacogenomics may have a potential to transform how traditional medicines are prescribed. In doing so, it can widely increase the patient outcomes and can also reduce the health cost. So in the future, you might see and how pharmacogenomics transform how the drugs are prescribed, and we might even see that each person having the same disease may be prescribed different drugs according to their genetic makeup. So this is the end of my presentation. Thank you for listening. And here are all the references that were used in making of this presentation. Thank you. Bye bye.